Yeah, thank you so much, Sonia, and thanks for the very kind invitation to and the opportunity to speak to all of you. I'm from a little bit different background than most of you guys. I think most of you are like more like biology and stuff. So I'm more in the physics and optics kind of background. But what I try to tell you a little bit about is a different kind of tool that we're trying to develop for microscopic and nanoscopic objects, whether these are more on the biology side, so cells, or whether these are like synthetic colloid stuff or like material science. And hopefully can give you like an outlook, what is like maybe coming in the next couple of years. And so the way I phrase this talk is basically centered around optofluidics. And if you ever heard about optofluidics, it's basically quite a large mixture of things, and it basically combines two different types of tools. So optics is on the one side, which can use, for imaging, for sensing, also for manipulation. And then on the other side, you have the microfluidics part. So yeah, where you basically control fluidic environments on a small scale. So if you heard about a lab on chip, organ on chip, they all kind of use a microfluidic environment. And so I'm approaching this topic a little bit different. So most of these techniques are using like pressure-driven systems and very microfabricated environments. We want to do everything with light and heat only, and that's basically the general idea. And so let's get into it. And so what is basically the central topic of it is how you can gain microenvironmental control. Yeah, because on one side you will need this to basically do manipulation, right? Because you want to move things from A to B. That would be the simplest thing. You want to steer things, or you want to do even more complicated tasks, such as sorting particles. Imagine you have a heterogeneous samples of different particle sizes, so different sizes of cells, or the cells can have different shapes, and you want to separate them. So this is kind of like the most traditional kind of task that you want to do in microfluidics. So you have a heterogeneous sample, and you want to select the ones you want and deselect others. But you can do other types of tasks, such as isolation. You want to identify, later maybe even do imaging and sensing of different specimen. And so there are, of course, different scales and different things that you can study. So on the nanometer scale, you maybe have more like proteins that you're interested in looking at, or nanoparticles. And if you go one scale up, basically, you can even look at cells and microparticles. But in order to do all of these different types of and quite complicated tasks, you need a certain environment, and you need certain types of tools, right? And so basically the other side of the coin is basically using the microfluidic part. And so microfluidics is great because it gives you quite a high control over these environments, but basically spatially shaping the way it looks like. You can have, for instance, a single channel, multiple channels. You can put pillars inside, which change the flows and then can be used for sorting. And so on one hand, you want to have basically, if you think even about lab on chip devices, you want to have multiple functionalities. So not just, maybe don't just want to do sorting. Maybe you want to also do isolation after, or then do an imaging part, or cultivate cells later on once you isolate it. And so you can make this quite complicated, but doing this in a single device is quite complex. As you can imagine, sometimes you require multiple layers on top of each other. And then on the other hand, you want to also handle a really broad range of specimen, right? You don't just want it to work for a single cell size because maybe cells are growing, and so they have different cell sizes, so they might not work with the same configuration. So the point I want to try to make here is that for microfluidics, you require a certain degree of flexibility and also reconfigurability, meaning that you can switch between different tasks. And so if you look at the production side, so to say, of these microfluidic chips, so traditionally you use now these optical techniques where you can either like 3D print in like a solution. Maybe the classical 3D printing of like a plastics or a polymer. You can do this also like in a kind of resin, and you can do really fantastic stuff. Think about the nanoscribe and all these kind of startups that now become really, really big. You can do direct laser lithography. All these fancy tools, they enable you to do like these microstructured environments. But of course, once you fabricated them, they are fixed, right? Whatever you have to do in terms of you have to change the size, you have to change the spacing between pillars, that's not possible anymore. And so we can look at these couple of examples. Imagine on the left, you have more like a microvell structure, which people use for cultivating cells, for instance. And so either you have all the different size ranges already included in your chip, but again, it's a fixed design. You can't change it after you've fabricated. So especially when you want to do prototyping and stuff, it can be quite time consuming because you have to go back to a clean room, fabricate from the beginning and test all these parameters. And on the right side here, for instance, this is one of the particle sorting. It's now over 20 years old, and it's called deterministic lateral displacement. I will come back to this later. That's why I want to mention it here already specifically. And the way it works is basically you have pillars of different sizes and different spacings, and simply by that, you can sort particles simply by their size. Okay, very simple principle, but of course, you're quite limited in the range of particles that you can separate once you have these fabric structures. And so I want to introduce now a range of tools which we call optothermal tools, which is part of the optofluidic kind of toolbox, so to say. And they're great because all you need is basically temperature. And temperature is really easy to achieve, right? If you use light, it's great because you can have it very localized, and all you need is basically an absorptive surface. Yeah, it can be a plasmonic surface, can be just an absorbing film. Imagine, for instance, gold absorbs in the visible length, wavelength range quite well. And so this has been now shown to do all these different kinds of tasks, and you can read, for instance, trapping, rotating, stuff like that. Here are a couple more of these examples of very specifically in the field. The way it basically works is that you somehow create a temperature gradient using light, right? So you don't use light directly to manipulate the particles, which is more like the optical tweezing, like a community. Here we really just use the temperature modulation by basically getting different types of temperature gradients and landscapes with which you induce fluid flows. And if you heat up a fluid, you might know that when you basically look also in air, if you heat up, you have these particles running around, you have these convective patterns. This is like one way of doing that also at the micro scale. You can get this buoyancy basically of your particles because the density of your fluid changes with temperature, so you can start moving fluids around. But you can use also different kinds of other effects like osmotic effects, so things that happen near the surface. Or if you really quickly scan a laser, for instance, you can get viscous effects that all depend on temperature. So you see that temperature is quite a neat parameter, environmental parameter that you can play with to achieve all these different kinds of manipulation. And so this is basically where I want to start with. So a couple of mistakes here, but just bear with me. So the idea is basically that we want to replace these microfabricated barriers that we've talked about so far with what we call an optofluidic barrier that we just basically create by increasing a temperature and creating these virtual flows that act as a barrier. But they're not in a sense like a physical barrier in that sense that you have something fabricated, something rigid. But it's just something fluidic, but it will act the same way. And so the way we do it is by, in our case, we have plasmonic nanoparticles, which we homogeneously spread on a surface. So these are, for instance, called nanorods. They are great because they absorb basically in the near infrared only. So if you want to use that for biological applications, it's great because that's basically in the optical transparency window of biological specimen. So you don't want to use visibles, you want to rather go to near infrared. So that's why they're great for. But in general, any absorptive surface will do. So you shine your laser light on it, it will absorb, and because it's locally confined, you also get a locally confined temperature field. Now, we can measure these in experiment, and you can also see it nicely in simulations. What will happen is basically you get a hotspot with a single laser beam, all right? And so now you can tune this, and this is the first parameter that we wanted to address, the tunability. So by just changing basically the incoming energy or the laser power that you put in, you can change the temperature gradient. So the more energy you put into the system, of course, the higher the temperature difference will be that you induce, and therefore you get a higher gradient. And this will be essential if you want to drive fluid flows, because the higher this temperature gradient is, the faster you will drive your fluid flows, the stronger is basically the interaction of your optofluidic barrier with your particle motion. And so the second part of it is because we can now modulate light in any kind of way you want. One of the techniques, for instance, using a spatial light modulator, don't need to understand how it works exactly is, but we basically can shape a beam in any kind of way we want by changing the phase of the beam. So we can have, for instance, just a single beam but focused also at 2 different surfaces. Or we can create really multiple types of beam, and that's what I'm trying to illustrate here on the bottom, is that we have now a microfluidic chamber where we have absorptive surfaces on both sides. And what we do is we focus a laser beam first on the first one, the first surface, and on the second one, which will give us basically what you see on the top, just from both sides. Or we can create any kind of other shape, and that's what we're getting to, is basically by spatially modulating this, we can create different kinds of what we call microfluidic actuators. So kind of like functional barriers that act with particles in different kinds of ways. And so if we now look at the simple case where we basically have just 2 heat sources from both sides, what we do here is we do particle tracing. So we have particles that don't interact with the temperature itself, but we just look basically at how the fluid flows are moving around. You basically see that near the surfaces, you have the osmotic effect. So it drives towards the hot regions near the surface. Then it goes along that gradient up. And of course, since mass is conserved, and eventually the flow has to come back, you get these recurrent flows from both sides, of course. And if you look at it basically from the side here, so that would be basically in the direction of the laser, you see that really these things are going towards the center. And so this we can now use to drive fluid flows and create barriers. And so just to come back, basically, for those that are more interested in the technical parts, how does it actually work? So when you heat up a fluid, there are many effects that can happen because you have different types of fluid parameters that you can change, such as viscosity, density, the entropy, and so forth. Why I want to highlight these 3 different ones, which is the thermoosmotic ones I mentioned, that's the one that's just near the surface. It's also the strongest in our case. So you can see we get on the surface up to 20 micrometers per second for a given temperature difference. I think it was around 10 or 20 Kelvin. You get the convective effect. That's what you're probably most familiar with. This is just basically going against gravity always. So it's a unidirectional flow. And then you have the phoretic effects. Phoretic effects is basically kind of like an osmotic effect, except that it doesn't happen on the boundary surface, but on the surface of the particle itself. And most particles that you will experience, but also bacteria and cells, they're kind of thermophobic. So they don't like heat too much, so they move away from it, which is why you see these drift fields to be going centrally outwards. And so the nice thing about this is, if you know the system really well, you can tune each of these parameters individually. I'm not going to show all of the data for each of those, but what I want you to show is that this system is really versatile, because the thermoosmotic effects basically are pretty constant, except you basically change the materials you use on your boundary. Convective effect basically depends on the volume and how much heat you can transport away from your microfluidic chamber, meaning that if it's very small, heat can go away very quickly. And in this way, you can basically suppress convection if you don't want this unidirectional flow field. But if you make it really thick, and thick I mean like 100 micrometers and thicker, you get a huge convective force. You can drive particles a lot along this direction. And the thermophoretic effect really depends on the particle. So in the end, you can use this even for particle sorting, because different types of particles will react differently with the field, and people have been using this for this kind of applications. All right. Let's come to the optofluidic barrier, so the interesting stuff. And what we basically do is here, we alternate our focus of our laser beam. And so we go one on one surface, then we move a little bit laterally, then you focus on the other surface. And we do this, and it kind of looks like teeth that are intercalating and forming this thermal barrier. And if you do that, and here's a video, if you look at it from the side, you have particles that in this case are just sedimenting by gravity, but you can also put a flow. They really avoid this barrier. So they come down, and then they kind of get stopped. You see this one on the left, and then it cannot pass through because there are these fluid flows that keep it from going through. And the only way it can go along the sides. And this way we basically build a wall. You see also the trajectories here in white. So all of these particles, they have to find a way around in order to pass through, pass besides this barrier. And if you look at it from the other side, you basically see that even some particles, they get repelled a little bit, right? So they are basically at these hotspots, so where the laser beam is, They have really strong flows. They push it away because they have this convective force again that goes against their movement, right? And then it works really well. All right, now we can change this configuration, right? I said we can change the laser beam and the profile of it, and definitely we can change the temperature profile. And with that, we can do all kinds of what we call actuators. And so the first one, so really basic ones here, is a deflector. So all it does is basically deflecting particles to either one side. In this case, we just show towards the right. You can kind of imagine it in microfluidics terms, like a channel that is inclined, right? And so this works really well. And here's a kind of an example with lots of particles. So you see they, of course, the ones on the left can go on the left, but the ones that were on top, they kind of bounce along these flow fields and then all move to the right. And we can do the same, of course, not just for a collective of particles, but we, in this way, because we can reconfigure it in real time by just using like a feedback between our imaging and our laser beams, we can individually address particles. So if you have a targeted particle, a targeted cell, you can address that by just tracking the particle's movement. And so in this case, we just move it basically simply to the right and to the left and to the right again. So kind of like playing ping pong. So this can be addressing particles really individually. Now you can do all kinds of other things. For instance, the splitters, it's kind of like this Y channel, which is very common. So you split the particle flow in two. You can have like a channel that becomes much narrower. We call this a merger, because you kind of like converge all the flows into one single one. And the cool thing is that we can also do fluidic trapping. So again, just as a comment here, that optical forces are do not play a big role here. They're pretty small in these cases. So this is trapping, but it's not working by optical forces. And the cool thing is here, because we have this U shape and particles are coming from the top, so it acts kind of like a micro pocket or micro well. And all we do is now we basically change the laser power. And if we change the laser power, we change the convective force mostly. So we use a little bit of this convective force. If we increase the laser power, the temperature gradient is stronger, and therefore we push the particle away. Then we decrease the laser power, and the particle comes back down. And so you have really a 2D control, because now you can imagine you can move that whole trap left and right as well. And with that, you can really control a single particle really, really well. All right. We thought we gonna take this actually a step further, and we thought, okay, now what can we do more? And we thought of these periodic arrangements of optofluidic barriers, which kind of like becomes like these pillar arrangements that I talked at the very beginning. And if you only have particles that come in between the two center ones, and then you have equal spacing, so just basically half of the distance of the next row to the other one, what will you get out of it is, if you think about it, this is an experiment that you have probably seen in high school or undergrad already. It's basically the Galton board experiment, which is the physical demonstration of a mathematical concept, basically meaning that the central limit theorem, that if you have enough statistics, you will get a Boltzmann distribution, a normal distribution out. And because particles will basically either go left or right, and they go left or right. So in the end, though, you will have most particles being at the center. And although this is working really well in experiments, it works well for different particle sizes as well, which is great. You see that, of course, the smaller the particles, the more you have the Brownian motion that becomes a bit more random. And the flow, the sedimentation speed is a bit smaller. But nevertheless, it works really well. So we can do now, basically, changing the spacing allows us to really modulate it towards the particle size that we're targeting, and still getting the same kind of effect out of it. And so if you now just slightly modify this pattern, and instead of moving it by a half, you just move it by a third. What does that mean? Is basically that on the right side, you're forming kind of like a larger gap, whereas on the left side, you have a really, really small gap. And now you can do particle size sorting with that. So bigger particles, they will not be able to pass through that smaller gap, right? But towards the one towards the right. And if you do that often enough, that is exactly the principle of this deterministic lateral displacement that I've shown before, where you had these pillars with a certain spacing. And what we can do here now is, and we demonstrated this, we can really separate particles. Of course, this is, still quite noisy in the sense that, not all particles will end up on the right immediately. But I wanted to show you again, maybe here, is that people have been separating these particles over like a millimeter range. What I'm showing you here is a field of view that is just like 100 microns, and you already get more than 50% separation efficiency. And the cool thing here is that we can do it for a really heterogeneous sample. We just show basically 6 and 4 micrometers here, but we can do it for all kinds. If you have really small particles, it works as well, because you get a convective part. All you have to do is now, instead of moving it downwards, you just flip the whole pattern, and you can separate them upwards, basically, because smaller ones, they don't sediment as fast, but the convective part is larger. And so it just shows that this is really, really versatile. And in simulations, you can see that actually even better. And so this is a great way of doing particle size sorting. I'm not saying that this is the way you should do particle size sorting. There are much, much faster ways of doing it. But what we wanted to demonstrate here is actually that it works, and that these barriers actually work or act as a physical barrier or as a traditional barrier that are microfabricated. The cool thing now is you can modulate these patterns, so you can have different heating spots and make the efficiencies even higher, just to show that there are different ways of improving on this.
Now, just to let you know, about 5 minutes or so, 4 or 5 minutes.
Yeah, I'm basically done. So
okay, yeah, no,
perfect. No, thanks for the reminder. Yeah,
no, take your time.
So that's the outlook. So where do we want to go from here? So now we basically have this proof of principle of it, and the idea is now that I've shown you all these different types of actuators, the different types of function that we can integrate. Now it's basically the time to integrate it all into a single device. Basically, you just need a large area, and you can create these spatial patterns by using this temperature engineering to create different types of functionalities that you can use for self-organization in cells, or other kinds of particle analysis and catalysis, so for chemicals or material sciences. Not saying it works 100% for all of this yet. I think there's much improvement to be done. And I know that this question is going to come up, so I'm going to take it away, which is, of course, temperature is going to be relevant for biological applications, right? What I want to stress here is, although we use quite high temperature ranges, up to like 40 Kelvin, so above room temperature, that would be like 60 degrees Celsius, that would be not relevant, that would be not applicable to biological applications anymore. But keep in mind that not the absolute temperature is what determines the functionality of these optofluidic barriers. It's actually temperature difference, right? So if you can play with this, and you can decrease the range. So if you have just 10 Kelvin difference, that would still be okay at room temperature. You could lower the room temperature further down, or the temperature of your sample. You can use higher temperature gradients. It will still work the same way. All that matters is basically, that's what I want to stress here, is the temperature difference. Yeah. With that, I want to do a bit of self-advertisement, because we're organizing an Optofluidic conference. I'm quite excited. It's the first one I'm organizing this year in Leipzig, Germany. It's going to be all about optofluidics, not just the optothermal stuff I will show you, also more traditionally on imaging, sensing stuff. So if you're interested, the website is up soon. So keep following me, or keep checking out the title of this conference. I would be very happy to see you there in person. With that, I want to just thank my funding sources, the people that have been involved at ETH. And if you want to read more about it, here's the link to the archive paper. It's hopefully online soon, the paper. Yeah, if you have any questions, very happy to take them.